wonderful to see so many people here, not just from the Arnold School, from all across campus. I'm Tom Chandler. I'm the Dean of the Arnold School, and today is a very special day. We have a very special guest visiting with us, Paerl, who Dr. Scott will tell you more about in a moment. This is our 15th Winona v. Vernberg lecture in public health. We try to alternate or change the public health topics of this lecture. It's our key lecture for the year. We do several throughout the year, but this one we try to elevate to our highest level with the highest quality of speakers and vary across the many fields or many disciplines of public health. And this year is environmental health year. Winona Vernberg was a very special dean to us. She was essentially our founding dean many years ago. She's been passed since 06, and I'm sure she would be proud to see how the school has grown and changed, and particularly the faculty and the students that we have that are really a part of her legacy here in Columbia. I'm delighted to see Jerry Odom here, who worked with Winona back in the day. Jerry was our long-standing provost at the university. This is the guy who actually made the mistake of giving me tenure. Thank you, Jerry. So anyway, we've got a great lecture today. Without further ado, I'll let Dr. Scott kick it off. Okay, thank you, Tom.
It is a great pleasure to be here today for this lecture. And I cannot thank everyone enough for coming out to hear what is going to be a great talk. We had a meeting this morning with Dr. Paerl with our South Carolina HAB network, with DHEC and all our folks addressing these issues. So really delighted to see everybody here today. I wanted to mention something about Winona Vernberg. Tom mentioned it. Tom and I both worked a lot with Winona and John. And at the one of the things John and Winona did is they put out 9 different textbooks between 1970 and 2000. And this was when we were still using typewriters. They were really always involved with pollution response in marine organisms. At the annual or semi-annual Society of Pollution Response in Marine Organisms meeting this year, we established the F. John and Winona B. Vernberg Award, the Pioneering Award. There are only 2 awards given. One is the Rachel Carson and this award, because they were indeed pioneers. And if you knew anything about Winona, she always liked linking environmental health and public health. And so when we take a look, if we take a look at the concept of oceans and human health, traditionally we have been focused on looking at how man's impact on the oceans can change ecosystem health. But we suddenly are now realizing that when we change the ecology of the system and change the ecosystem health, that can affect us, our human health. And so that is, this is what we call a One Health concept, and we're certainly talking a lot about this in classes, lectures, and courses. So one of the things that we're delighted today, our guest speaker, Dr. Paerl, is from UNC, and you'll hear a little formal about his credentials here in just a second. But our center here at USC, we're part of 3 other centers on oceans and human health. And so Dr. Paerl is a member of the Great Lakes Center. And so we're really delighted to have him here today to talk to us about an emerging health issue, and that is harmful algal blooms. And so just to give you a little bit real quick about our center, we have an administrative core of 4 projects, and these are the folks that lead those: Sean Norman, Brian Brooks, John Perry, Saurabh Chatterjee, and Dwayne Porter, and then Paul Sandifer and I run the administrative part of this. And so we're really delighted to have one of these centers to help address these kind of issues. Now, I want to tell you a little bit about Dr. Paerl. First of all, he'll tell you in a minute, he knew the Vernbergs. So it's really nice to have a speaker who actually knew Winona and John. And he is the William R. Kenan Professor of Marine Science at the University of North Carolina, Chapel Hill. He's located on the coast in Morehead City. And just a couple of things, highlights that he has been involved with. his expertise is in really understanding how ecological health responds in terms of ecosystem health, how they respond to the increased occurrence of nutrients and climate change, and all these things that can change and cause harmful algal blooms and affect human health. One of the programs he runs, how many people have been on the ferry boats in North Carolina? You may not have realized, but there have his monitoring system on these boats called Ferrymon, and also his Modmon program are all big parts of what he has been running up in the North Carolina area for years now. And in addition, he is funded by NSF, NIEHS, NOAA, Sea Grant, EPA, Department of Defense, and a number of state agencies in North Carolina. And his lab at the Institute of Marine Sciences, Microbial Ecology and Nutrient Cycling Lab, is very prominent. And he holds joint appointments in the Department of Marine Science and Environmental Sciences and Engineering and the Department of Biology and Ecology curriculum. His formal degrees are from the University of California, Davis, both his BS and PhD in ecology, I will point out. And then he has won several outstanding awards, the G. Evelyn Hutchinson Award from the American Society of Limnology. The big one here, I think, Eugene Odom would be proud, the Odom Lifetime Achievement Award, the father of ecology. And then the Chinese Academy of Science as a fellow, visiting Geophysical Union fellow, and then also visiting professorship in Netherlands through the Royal Academy of Arts and Sciences. He is a member of a number of editorial boards. I'm not going to read all those. I just wish I could be on one of those boards. So I'm very unbelievable expertise in microbes, ecology, oceanography, you name it. Dr. Paerl has done it. And I counted the publications in his CV. It took me about an hour, but there were 498. And just to let you know, the last year he did 15, and he was first author, I believe, on 5. So he hasn't slowed down a bit. So without any further ado, I am really delighted to introduce Dr. Hans Paerl to talk to you about mitigating harmful cyanobacterial blooms in a hotter, hydrologically more extreme world. Dr. Paerl.
Thank you very much, and it's particularly, well, an honor for me to speak at this symposium, I guess, because I knew the Vernbergs very well, and they really clued me in on a lot of stuff when I was just a beginning professor at the Institute. So it's really nice being able to come back to a place that I feel very familiar with. And also, some of my colleagues who I've worked with in the past are esteemed professors here, including Alan Decco and Jay Pinkney and Tammy Richardson. So it's like a homecoming over here. All right. Well, I'm going to talk to you about cyanobacteria. They're my favorite organisms. They've been around the longest of any photosynthetic organism, so we should be thankful for them because they invented oxygen-evolving photosynthesis, and that really got higher forms of life started on Earth. And they've been around for over 2 billion years. And I wanted to point that out because, 2 billion years, lots of things have happened on the Earth. Geological changes, much hotter periods than we're having now, even though it's getting hotter, more extremes, and they've experienced all this. So their playbook is very deep. And this is something that I think we're still really coming to grips with in terms of understanding these organisms and why they're so successful. Even now, in fact, they can thank us for being successful in terms of pollution and eutrophication problems. So I just wanted to sort of start off with that, just to give you a little bit of background on these organisms. Okay, I'm going to talk to you about lots of different places where cyanobacteria have been successful, and also how they have really dealt with changes on Earth, including the Anthropocene, the human period of human intervention, and climatic changes, And I wanted to acknowledge many other folks that the list is so long that I couldn't put them on the opening slide, but lots of people in the US, Canada, Europe, Asia, China, South America, Africa, that have all contributed to our knowledge of these interesting organisms and how they've sort of proliferated on Earth and are now really a big problem that's so successful. Okay, a lot of things most of you already know. I think I can go away from the microphone since it's not working anyway. And I think I'm pretty loud, right? So you guys can hear me in the back? Okay. So what are the drivers of cyanobacterial dominance? And I might mention cyanobacteria are bacteria, they're not algae. So that's an important thing. If you look at them under the microscope, they look just like bacteria, except they have pigments in them that algae have in, like chlorophyll and some of the accessory pigments. So they're a very unique group. This again has a lot to do with their success. So what do we know about drivers of cyanobacterial blooms? And the black stuff is stuff you should probably already know. Okay. Human activity brings with it nutrients, particularly nitrogen and phosphorus. Those nutrients are then utilized by cyanobacteria to form blooms. But also water use and hydrologic changes are part of the formula that makes them successful, particularly our intervention in aquatic systems like building reservoirs and dams and things like that. And we'll come back to that. And climate changes. Like I said, their playbook is very deep, but they're particularly adept at taking advantage of warming and changes in precipitation. And let me tell you, I know everything about how much precipitation has changed over the last 20 years because in North Carolina's coastal systems, 6 out of the 7 wettest tropical cyclone events have occurred in the last 20 years, and that's based on a 120-year data set. So things are changing, and you've noticed it here in South Carolina too. They can take advantage of that. And one thing we all know about cyanobacteria is blooms are intensifying and spreading. And that's not just only because we're studying them more. There's a long history of that, particularly in developing countries. Okay, why are we concerned about cyanohabs? And this is probably going to be the only slide that really deals with what you guys are really good at, the toxins and all the human impacts and animal impacts. They produce these secondary metabolites that can be toxic to all sorts of life forms, particularly animals and higher animals. We don't really know why they produce these toxins, and in fact, to the cyanobacteria, they're probably not toxins at all. They're part of their growth strategy, that most of them are inside cells, so it must have something to do with their metabolism and their lifestyle, okay? And if any of you want to do research on this, this is a great topic to go into, is to understand why they actually produce these toxins, okay, or toxic substances. And my friends assure me that this is only the tip of the iceberg, that some toxins that we know a lot about, like microcystis, there are many other secondary metabolites that are yet to be discovered that may or may not be deleterious to other life forms. Okay, they also cause hypoxia and anoxia, which is the oxygen depletion in bottom water. So when these blooms die, there's a lot of biomass that goes down to the bottom, and that consumes a lot of oxygen, which can be detrimental. Odor and taste problems are often associated with cyanobacterial blooms. And then, of course, the human impacts, the loss of drinking water, recreational use, fishing, sustainability, etc. So lots of negative things here with the toxin aspect. Okay, I thought I'd throw this slide up here because often we talk about these organisms and you never get a chance to look at them. So I'm just showing you the 3 major groups microscopically that we're concerned about. There's the I'm going to need to use your pointer after all. It's okay. All right, so the top group are the coccoid, solitary, or colonial cells. And the genus Microcystis is in that. And everybody knows about Microcystis because it's one of the dominant bloom formers that we see in many lakes. And here you see the colonies that are present. Most of these do not fix nitrogen, and we'll get back to this nitrogen fixation aspect, but that's another trick that cyanobacteria learned. They can actually, some genera can use atmospheric dinitrogen gas and convert it enzymatically to ammonium. So that provides nitrogen from the atmosphere, okay, to get around nitrogen limitation. These guys don't fix nitrogen. They're dependent on nitrogen that's already in the system, and we'll get back to all that from a management perspective. Then the second group are what we call filamentous non-heterocystis. These are filaments of cells stacked on top of each other. They can form colonies, but also solitary, and Limbia, which I think many of you know about because it's a problematic organism in lakes here, and oscillatoria are in that group, and most of them don't fix nitrogen, but some of them do. And then the last group are the filamentous heterocystis guys. They have these special cells called heterocysts, and in the heterocysts is where the nitrogen fixation or fixing apparatus is. And this is a really clever adaptation because these cells are producing oxygen by photosynthesis, and yet they produce cells every now and then that are conducting a process, nitrogen fixation, which is oxygen inhibited. So this is, a pretty sophisticated evolutionary step that they've taken. So these guys often take advantage of systems where there's a lack of nitrogen in the system already. Okay? All right, so I'm pretty dangerous with 2 of these. So one of the things I wanted to point out is that, we used to think of cyanobacterial blooms only being a problem in freshwater systems. If you go back to the historical books, etc., they always talk about lakes and rivers and reservoirs, but they are expanding into estuaries. This is Brazilian lagoons, St. John's River in Florida, just down from Jacksonville. Anybody know this place? People go there to party and have a good time.
They don't know his name.
Lake Pontchartrain, just north of New Orleans. Here's a big Microcystis bloom on Lake Pontchartrain, which is a brackish system. And then they can get out into coastal seas, like the Baltic Sea, for example, all the way up to fairly high salinities. These are all blooms on the Baltic Sea. And, the salinity tolerance for some of these groups is actually quite good. If you look at salinity, this is the increasing salinity on these graphs. And we look at the different bloom formers, you see some of them don't do, well, some of them are pretty adaptive up to, say, 10 parts per thousand, which is a third of seawater. But some of them actually can grow all the way into seawater salinity, like Nodularia and Anabanopsis. And so the take-home message here is that salinity is not necessarily a barrier to these blooms expanding. And it may very well just be a nutrient thing, so that, as our coastal waters get more nutrient enriched, the cyanobacteria may track that and move out into some of our estuarine and coastal systems. That's just a kind of a, word of warning about how adaptive these blooms can be. Okay, I'm going to talk an appreciable amount of time about hydrologic variability and warming because we know what's going on. And how do the cyanobacteria take advantage of these types of events? We know, for example, we're getting more extreme tropical cyclone events, as you can see in the Atlantic here, with more rainfall, which is leading to big flushing events. But once those flushing events have dried out, then you have a system which essentially has the nutrients put into it. And if that's followed by a drought, then you kind of have the perfect storm scenario. You got the nutrients that have come in, followed by a summer drought, or a drought at some point, which allows the cyanobacterial blooms to build up. They're not getting flushed out, but the nutrients are in the system already. So they can take advantage of that. And we know things are warming up. This is the North Atlantic temperature anomaly. Okay. So I'm just going to show you a few examples of where cyanobacteria have, in fact, gone from the freshwater into coastal systems. And in some cases, they've gone into coastal systems and they end up at high salinities, lysing and dying, but that's not the end of the problem because the toxins can then be released into the saltwater system. And this is some work by Rafe Kudela and others at UC Santa Cruz that have shown that sea otters, which like to eat shellfish, and the shellfish is contaminated with the toxin because they're filtering the water, can be adversely affected by blooms that start up in the freshwater component of these systems. So there is that connection then going on with the coastal system. North Carolina, I think everybody knows the Albemarle Pamlico Sound system, the second largest estuarine system in the country. Well, we have some components of that system, like the Albemarle Sound, which are brackish, and they're experiencing essentially an invasion of cyanobacteria through the tributaries into the main sound as we're getting more nutrients put in, and those nutrients are being pulsed by storm events into the system. And these systems are big enough so that they can capture the nutrients without everything being flushed out into the ocean. So this essentially is a big incubator for the blooms that have been coming into our sounds. I don't know why that doesn't show up, but I'll tell you what's in this box. Another analog to this is the Mississippi drainage area into the Gulf of Mexico, where we've had tremendous floods and an increase in floods that has essentially created these freshwater coastal habitats. And so the diversions now that used to keep the freshwater upstream have now given way and allowed freshwater to come in, and with that, we've essentially had a spread of cyanobacteria into the coastal waters of the Gulf of Mexico. And those have been shown to be toxic as well. Same players as we see in North Carolina. And then, I think many of you are familiar with this scenario, Lake Okeechobee. Huge lake built as a flood relief reservoir, but also a water supply at the same time. And of course, with the increasing tropical cyclone activity and more rainfall, essentially the system has overflowed, and they've had to release water out of that through the Caloosahatchee, going out here to the east coast of Florida, and also down towards Sanibel on the west side. And here you see the scenario. The blooms are here. This is the perfect incubator. It's being transported into the coastal systems, leading to lots of problems in the estuaries. And the blooms don't necessarily die right away. Some of them get replaced by other more salt-tolerant species. But even Microcystis, which is the dominant bloom former here, can live at several parts per thousand salinity, up to maybe 10 or so. So this has really transferred the problem from a freshwater system into a coastal set of environments, and it's caused a lot of havoc in Florida, to say the least. Okay, temperature. Increases in temperature play a big role in the expansion of these cyanobacteria, because cyanos like it hot. Many of you are old enough to know that famous movie, so I'm using that term. And if we look at the growth rates of the different algae that are competing in most freshwater systems, we have the diatoms, the dinoflagellates, the chlorophytes or green algae, and cyanobacteria, and we look at their growth in relation to temperature. You can see that the cyanobacteria have the highest, their optimum is at high temperatures. So as things get warmer, and they warm up earlier, it gives them a jump up on the other algae in terms of the growth rates. So they're taking advantage of warming. And we know systems are getting warmer because there are long-term data sets that show that. In the Netherlands, for example, this is a paper by Geoff Scott and his group. They've shown that in the Epilimian, the upper layer of lakes in the Netherlands, the temperature has steadily increased. There's no doubt about that, probably about 2 degrees or so. And that not only creates higher temperature, but it also stratifies those systems more strongly, because the warm water is light, overlaying the dense cold water, so that strengthens the stratification. And that plays into the cyanos playbook, because they like to be up at the surface, and they form these surface-dwelling blooms that many of you have probably seen in lakes and reservoirs. So it benefits them in 2 ways. And then if we look at Lake Erie, and this is data from George Bullerjean's review, and you have to be a little creative with this data, but you can see that overall the temperatures have also increased in Lake Erie. There's a lot of scatter there because it varies from year to year, depending on whether you have a warm year or a cold year. But overall, temperatures are increasing. So the window of opportunity for the cyanobacteria has increased as well. And this is data from a paper by Saar and Kosten from the Netherlands, where they sampled, let's see, how
143 lakes from the tip of South America all the way up to the Arctic Circle. And what they did was they plotted the biovolume, the amount of biomass that cyanobacteria represent in the total algae component of these systems. And you can see that as the temperature goes up, the relative dominance by cyanobacteria increases. And then if we throw nitrogen into that as another correlate, you can see that with nitrogen enrichment, the relationship is even stronger. So as the world is getting warmer and more nitrogen rich, we're seeing cyanobacteria take advantage of that. And we'll get back to the nitrogen story in a minute here. Okay, hydrologically things are getting more extreme. And I think I don't need to tell anyone in South Carolina that, because you're well aware of the flooding that's been associated with tropical cyclones. North Carolina, of course, we have the pigs learning how to swim. Not so successful. And here's again the increase in tropical cyclones. So things are getting more extreme. And this is data from a vertical profiler. So we put these vertical profilers in our estuarine waters in places, in lakes. And what they do is they collect data from the surface to the bottom every 10 minutes. It's just a set of sensors that goes up and down in the water column every 10 minutes. I think there are some actually deployed in some of the lakes in South Carolina too. And so we're looking here at temperature, salinity, dissolved oxygen, chlorophyll, and turbidity. And we had the good fortune, and in fact there's a little side story here. This was a project that I conducted as the only biologist in a group that was mainly physical oceanographers that had all this equipment out there. And they saw the storm coming, Tropical Storm Hannah, and they were ready to pull the equipment out. And I said, no, we don't want to do this. This is going to be the most interesting part of the story. So I had to promise that I would pay for the equipment if it got swept out to sea. Unfortunately, it didn't happen. So, Tropical Storm Hannah came along. And here you can see the wind field. And here is temperature. Everything becomes homogeneous because the water column is getting mixed up. Okay, so temperature is uniform, salinity uniform, DO. You can see there was a bloom here before the hurricane or the tropical storm. And it's really nice. You can see the algae migrating up and down, by the way, on a diurnal basis, which is really neat data. And then the storm came along. And look at what happened. The bloom actually got bigger after the storm came through. And here's the turbidity. So that tells you all the suspended sediments and things that are moving up and down in the water column. So the interpretation of this is that as the storm, mixed everything up, it also mixed nutrients back up into the water column from the sediments, resuspension. And the algae quickly took advantage of that and formed even bigger blooms afterwards. So they're well adapted to storms too. The storms don't just wipe them out. They can take advantage of it, come back. And in China, on Lake Taihu where I work, I've been there for 13 years, so I've been there through storms, high water, low water, everything. And what we did was we looked, this is Mango and Xu et al. We published a paper on this in 2014. We looked at chlorophyll from space. This is Lotus-derived chlorophyll, remote sensing. And you can see that in years where you have a typhoon comes through, the chlorophyll is always higher afterwards. And in years where we don't have the typhoon, the lake just becomes more nutrient-starved, and ultimately the bloom dissipates. So it's a very similar story to North Carolina. They call it typhoons, we call it hurricanes or tropical cyclones. But the response is very similar to these events. Okay, now I don't want you to get too, this looks very complicated, but I'm going to talk you through this hopefully. So, of course, we have to really deal with interacting effects of nutrients, because that's really the controllable factor in all this, with these climatic drivers. And in Taihu, the data are very good for a long period of time, because the Chinese, this lake actually has been kind of it's a very famous lake in China. It's 2,300 square kilometers, and it's revered in China. And the lake was, well, I'll tell you later about the history of the lake, but there's a good data set going all the way back to the 60s, and you can see that the wind speed over Taihu has decreased. And that has to do with large-scale changes in ocean circulation and wind patterns. You can see it's very consistent. The wind has decreased. Okay, the temperature has increased, particularly at nighttime, and rainfall has increased. Okay, so what's happened is the water level has increased in the lake, the wind speed has dropped, and the water column becomes more stable under those conditions. Okay, and the stability benefits cyanobacteria because they can just migrate up to where they want to be without it being mixed in. And the next slide, and this is actually a picture that I took last year of the surface of Taihu. So you can see here that there are also nutrient changes going on in the lake, and nitrogen has actually gone down in response to them starting to treat wastewater for nitrogen. And by denitrification, the lake is losing nitrogen. So this is a good sign. Phosphorus hasn't done much because phosphorus is hard to get out of a system. Once you get it into, there's no gaseous form. So it's essentially trapped in the system. So you would think that chlorophyll should go down in this lake. The bloom should go down because nitrogen is going down. But the opposite has happened in the last few years. And we think that's because warming is leading to more cyanobacterial production. They're more efficient at the high temperatures. The mean wind speed is decreased, favoring the blooms. The lake level is increased, favoring stratification and also denitrification, which benefits from a stable water column. Denitrification is the conversion of nitrate back up to atmospheric nitrogen, so it's a loss mechanism for nitrogen from the lake. Phosphorus has increased probably because of this increased stratification and more stability in the system. The bottom line is that despite all this, the chlorophyll has gone up. And we think that that's simply because the biogeochemistry has favored, essentially, these blooms in the system, the climatic conditions. So the Chinese have been asking now, well, why are we reducing nitrogen if we're not getting a benefit out of this because of the blooms, or reducing the blooms? Well, we did some earlier work using dilution Bio Assays, where we actually dilute the nutrients to see where the threshold is in terms of nitrogen controls. And it turns out that really what's happened is that the nitrogen has been reduced, but it hasn't met the reductions that are really needed to control the bloom. And with climate change acting to enhance the bloom, the bar is kind of set higher now. Okay, so from a management perspective, this is a problem, and we're facing that too. We're having to reduce nutrients even more in a warmer, hydrologically more extreme world. So, we're saying stay the course with nitrogen because you're not going to be able to reduce phosphorus very quickly in this lake. I mean, phosphorus inputs have to be reduced, that's for sure. But you're not going to see the actual reductions in phosphorus that quickly, because it's an element that does not have a gaseous form that can be released. Okay, so this is the whole complicated issue then. We've got the nutrients coming in. We have climatic drivers that are warming up the surface waters, creating more stratification, benefiting the blooms. If you have this high flow, low flow scenario where you get the nutrients put in and then you have a drought, well, then you create essentially the best conditions for the bloom to form and dominate. All right, now we're going to talk to you about nutrients and what can we do about all this from a nutrient perspective. This is Taihu from the air. It's 2,300 square kilometers. It's a huge lake. It's bigger than Lake Okeechobee, just to give you a little bit of a perspective there. And this lake has changed from a diatom-dominated lake. So diatoms are algae we like to have in the system. They're good in the food web. They don't cause problems by and large. This lake went through a state change as these cities were developed around the lake. These two cities, each Wuxi and Suzhou, they now have 7 million people in each city. And when I first started working here, they only had about 1 million people. So over a little more than a decade, the population has increased 7-fold. With that has come more wastewater, obviously, coming into the lake and more agricultural development in the basin. In the mid to late 80s, this lake went through a state change. All of a sudden, it went from a diatom-dominated lake to one with cyanobacteria. So it went over that threshold. We're now cyanobacteria dominant. So, of course, the Chinese want to address this problem and do some mitigation. This is what the lake looks like when you're driving a boat along. It's sort of like driving in very heavy whipped cream. The boat actually slows down as you go through the bloom, and that bloom is about, it can be like a foot thicker or even higher in the lake. If anybody knows something we can do with cyanobacteria, we can make some money together. Just come on over to China and we'll do some work there. Anyway, big state change and a very high demand to do something about this. And here's the historical increase in nutrient loading. This is total nitrogen, total phosphorus. You can see both of them have increased over time in the lake. Okay, so in a nutshell, here's the problem with the lake. If we look at nutrient data, this is total nitrogen over total phosphorus. Those are the two major nutrients. And you can see this goes through these wild gyrations, but they're repeatable every year. So in the springtime, when there's a lot of runoff, bringing fertilizers and other wastes into the lake, you see the TN to TP ratio is very high. It's often above 60 or so. And then in the summertime, it gets very low. And this is what we call the Redfield Ratio. This is the line at which we like to have balanced growth in systems. So if a system stays to this line, we usually don't have water quality problems, particularly if the nutrient levels are low. And you can see it just way above that in the spring, way below it in the summer. So from that, we hypothesized that maybe from a nutrient limitation perspective, maybe this lake is phosphorus limited in the spring because there's so much nitrogen there. Okay? And then it drops down below that line in the summer and becomes nitrogen limited. So we tested that using Bio Assays, which are simply very simple experiments where we take a bunch of containers, and in some of them we don't, and these contain lake water, by the way, just sample from the lake. And then we add, the controls don't have anything added to them. And then to some of them we add nitrogen, in this case it's nitrate, some of them phosphate, and some of them N and P together. And if you look at the response in terms of chlorophyll, which is the indicator pigment for how much algae there is in the system, you can see in the spring, here's the control, here's the nitrogen addition. This is the initial lake water, by the way. Nitrogen addition. Phosphorus addition, you can see phosphorus stimulates a lot because of that high nitrogen level. But then it reverses itself in the summer and fall with nitrogen being more stimulatory than phosphorus. And here's the next year. Here's the spring phosphorus stimulation. And then here you can see the nitrogen stimulation. So the lake is what we call co-limited. Both nutrients are important. They're just important at different times of the year. Okay? But if you actually add both of them together, you get the most at any time. So what this is telling us is that both nutrients are probably important, ultimately from a control perspective. And if you're someone who doesn't like to look at data, this is my politician data slide. So you can see this is the control. So, no nutrients added. 100 milliliters of tide filtered onto a glass fiber filter. Here's nitrate addition. Here's phosphate. Here's N and P together. Okay. How many people believe in this audience that both nutrients are important, looking at this slide? All right. Good. This is what I usually ask the politicians too. And they feel really good that they're right about it. All right. So the next question is, okay, we believe you, nutrients are important in Taihu. How much do we need to reduce the nutrients in order to get down below that bloom threshold? So to answer that question, we do what are called dilution bioassays, where instead of adding nutrients, we dilute the bioassay with a solution that's very much like the lake water, but it doesn't have the nutrients in it. Okay, we add everything but the nutrients. And then you get down to a certain point where you can see that you meet the control, where there's no stimulation. And then we add back the nutrients individually, nitrogen and phosphorus. Okay? So from that, you can then see what the dilution level is at which you could effectively shut down the bloom. And for phosphorus, it ends up being about 30 to 50%. For nitrogen, even more. Those numbers are big. But they're big. But if you go back to the historic increase in nutrient loading, they actually kind of match the increase that we've seen in nutrient loading. So this is telling us two different ways how much we need to reduce the nutrients to try to get down to that bloom threshold. And I know these are frightening numbers, but you know, I think this is really the price we're having to pay for not having paid attention to enriching these systems with nutrients to begin with. And you know, in China, I think the reality is setting in as well, and I think we've actually had a pretty positive reception to our work there. And they're hard at work now doing advanced wastewater treatment, best management practices, buffers, vegetative buffers, artificial wetlands, et cetera, to try to reduce these inputs in both N and P. So the next question is, well, are we just looking at a really strange system, or is Taihu representative of other systems, including Lake Erie and Lake Erie and other places? And I'm just going to quickly take you through some of those places. So it used to be that in Lake Erie, you know, they had a big problem in the 60s with these blooms. They then treated wastewater for phosphorus removal, and lo and behold, the bloom went away, but it's come back in the mid-90s, okay? Despite the fact that wastewater is all treated for phosphorus, and we're doing a good job with phosphorus, but nobody's paid any attention to nitrogen. And in the meantime, the blooms have come back, and this is the western part of Lake Erie with the Maumee River, which is a big contributor of nutrients, big agricultural watershed where they're growing all those biofuels that we're using, you know, that we're putting into our gasoline and stuff. Okay, so with that, there's come a lot more non-point source loading, and that has replaced the point source issue in Erie. And these blooms are all dominated by Microcystis, which is a non-nitrogen fixer. So it needs nitrogen to grow, okay? And remember, the phosphorus was reduced, but most of it's still in the lake because it hasn't left the lake. So the phosphorus is merely cycling around in the lake. It's the nitrogen that's really determining how big that bloom is going to be now in the springtime, okay? So again, both nutrients are really important. If we go to experimental lakes that have been manipulated by adding the nutrients individually, this is mainly from Canada and Sweden and Northwest Territory, Sweden, etc. You can see that actually nitrogen by itself tends to stimulate more than phosphorus, although there are some exceptions. But N and P together give you the most. So this fits the Bio Assay paradigm very nicely. So we published a paper back in 2016 called Takes Two to Tango. It's in Environmental Science and Technology. I'm glad to give you the paper. Anyway, this is the result of the, we even have a t-shirt. And whoever asks the best question gets a t-shirt. All right, and then if we go to lakes all around the world where these bioassays have been done, and where there are water quality problems that involve cyanobacteria, most of them show N and P stimulates together more than each alone. And this is a pretty exhaustive data set. Okay, just very quickly, estuaries. We don't want to forget about estuaries. So this is some of Tom Fisher's data from Chesapeake Bay, and he plotted these bioassay results over time, so from January to December for a year, and then the distance from the main river coming in there from the north, Susquehanna River coming in. So this would be Norfolk over here, and then Susquehanna up in there. But you see a very similar pattern to what we see in Taihu. Phosphorus tends to stimulate in the spring, but nitrogen stimulates during that bloom season in the summertime, and then it goes back to phosphorus in the fall. And light is limiting in the winter because there isn't much light. Okay, so you got nutrients limiting for most of the year, but light actually limits in the wintertime. And in North Carolina, we get a very similar situation. This is the Neuse Estuary where we work. Control, nitrate, ammonium, organic nitrogen, they all stimulate relative to the control. No stimulation with phosphorus, but if you add the two together, you get more than. I know this is starting to sound like a broken record, but I want to just make sure that that we get this dual nutrient situation across. All right, so this is some data from Sweden. You know those practical-minded Swedes. They really have taken this all the way to the management level. And they have problems in their fjords. So these fjords come into the Baltic Sea, which is brackish. And this fjord is downstream from Stockholm. And the good thing about, they've had bad bloom problems in this fjord, but the good thing is there's only one major source of nutrient input, and that's the wastewater treatment plant upstream. So from an experimental perspective, this is the perfect place you want to work at, because everything is all coming from one place. So, Ragnar Elmgren and Ulf Larsson decided, after they reduced phosphorus up here, they really had no benefit to the bloom. The bloom still kept on going. So they started to think about nitrogen as well. And so what this slide is supposed to show is that phosphorus was controlled already very early on in the game over time, but nitrogen was allowed to increase, so it went way up. And they convinced the wastewater treatment plant operators to start denitrification to reduce nitrogen. So here are the times when the nitrogen was reduced in the system. And hopefully the next slide shows you good. Okay. So here are those arrows for the nitrogen reduction. They were reducing phosphorus all along. And here's the nitrogen reduction, the inorganic nitrogen. You can see it was very effective because that was the main source. And here's chlorophyll, the indicator of biomass. And you can see very nicely that when they reduced nitrogen here, they also reduced the chlorophyll and got below the bloom threshold. And then, being good scientists, they said, well, let's just stop the denitrification for a year and let it go back up. Okay, so the nitrogen went back up, and look at the chlorophyll back up. So this shows very nicely that nitrogen could be a very important controlling nutrient in this scenario as well. And the really nice thing about this data is that they use that data to show the critical threshold for loading of nitrogen in the system. So as long as they kept the loading of nitrogen below 400 tons per year, they could get below that bloom threshold. This is chlorophyll again. If it went over that, the blooms would proceed. So this is a really good example of using basic data, monitoring data, to formulate a management strategy for a system. The advantage they had is that they only had one source of nutrients. And in North Carolina, South Carolina, they've got lots of sources. So that's the caveat to that story. Okay, so the question comes up, if we reduce nitrogen, are we just changing the system to allow these nitrogen fixtures to come in and fix nitrogen? And we're just moving from one bad habit to another, one bad habit to another. Okay? So that question is a valid question, and we tested that in Taihu by doing these mesocosm bioassays. And all we need to do here is look at the phosphorus addition. So if we added phosphorus to the system, that should stimulate the nitrogen fixers, because we're just adding a lot of phosphorus, the system's nitrogen starved. And if you actually look at the total nitrogen, this is all the nitrogen that's accumulated in the bioassay over time, there's no increase. And that's because nitrogen fixation, even though it might be going on, it's quantitatively not an important process to bring nitrogen into the system. Okay, so this is really important to realize, is that the systems become nitrogen limited, and they stay nitrogen limited even when the nitrogen fixers come into to replace the non-fixers. So why is that? And if we go to the enzymology of nitrogen fixation, here's the working part of it, the dinitrogenase reductase enzyme. That's reducing nitrogen gas to ammonium. Okay, and ammonium is a biologically available source of nitrogen that can then go into protein synthesis and growth. Okay, and here's the phosphorus connection. Okay, you need ATP to drive this process, so you need more phosphorus to keep it going. So, if this system was only reliable on the amount of phosphorus that's in the system, well, then you could control the N fixation rate by adding phosphorus. But it turns out that N fixation is a lot more complicated. This is the way it always works, right? When we do an experiment, it's always more complicated, and there are other things going on. And it turns out that this process is very energy demanding. to take up a molecule of dinitrogen and reduce it to ammonium is 16 times more expensive than just taking up ammonium directly. Okay, so it's expensive energetically. So you have to have energy to put into this process. And in cyanobacteria, that's provided by photosynthesis. In bacteria, through the use of organic compounds. But in either case, you have to have enough energy to drive this. So energy might be a limitation there. Iron might be a problem because if iron is not available, it's needed for the components of the enzyme system. And most importantly, I think, is oxygen. And we always forget about oxygen. You know, we're just sucking it in and out all the time, but we're not really thinking about oxygen very much. But it turns out that oxygen inhibits this process. So if there's a lot of oxygen around and it can get into the cells, diffuse in, it could also reduce the nitrogen fixing potential. And when you actually look at a bloom, like the one in Taihu, that can produce, say, 200% saturation of oxygen, you can see that if it falls on this curve, which is determined in the laboratory with Anabina, you see most atmospheric O2 would be about here, the equilibrium between atmospheric and dissolved oxygen is here. If we start to look at supersaturation, you can see the rate of nitrogen fixation. This is determined by a process called acetylene reduction, falls below the optimum. So we actually have data that shows that under the kinds of conditions that these blooms exist in with high oxygen, the nitrogen fixing apparatus just cannot provide enough nitrogen to support the growth. So what that means then is externally loaded nitrogen becomes really important because it fills that gap to optimize the growth of the cyanobacteria. And if we look at whole lakes and look at the flux of nitrogen going in and out, so here's the input, nitrogen fixation, here's the loss, denitrification. You can see that in most eutrophic lakes, it's negative, which means more nitrogen is leaving the system than coming in. So what that means is more nitrogen inputs will accelerate eutrophication because it makes up for that deficit. Okay? And so, from a management perspective, we've got to get more serious about nitrogen. Okay, and this is my nitrogen soapbox part of the talk. If we look at this conceptually, it looks like this basically. So we have the nutrients coming in. We have orange is the phosphorus, black is the nitrogen. It supports primary production, and as long as these nutrients are available in enough loads, you'll develop the blooms. The nitrogen fixers are in that bloom, so they're fixing nitrogen. But the bloom is producing a lot of organic matter that's then sinking down into the bottom waters where denitrification is occurring, which leads to the loss of nitrogen. So this is kind of a never-ending scenario. The systems just remain very nitrogen hungry, even when they get more nutrients to come in and support the growth. Okay, so again, it argues for controlling the inputs of nitrogen, which is really the key thing here. All right. So, believe it or not, this is my last slide. So what can we do about all this in today and the future? Well, I'm hoping that I've convinced you that both N and P are important. The old paradigm in limnology was just worry about phosphorus, don't worry about nitrogen. Okay, that's changing. It's changing largely because of our manipulation and the whole Anthropocene, basically. There's another good reason, by the way, for reducing nitrogen upstream in lakes, and that is as the waters make their way to the coast, we know those coastal waters are nitrogen limited too. So if you allow more nitrogen to get into the coastal zone, you're going to enhance the production and the potential for blooms down there. The blooms thresholds are probably system specific. So that means the reductions probably have to vary depending on the system that you're working on. But in most cases, we found that we need at least a 30% reduction in both nutrients. This is just the facts, you know, as they say in Washington. But so salinity is not a barrier. Salinity is not a barrier, and we know that now from experimental work that's been done and all the genetic diversity that's out there to allow scientists to come in. We may need to reduce those inputs even more in a warmer, stormy world because blooms like it hot, but they also like extreme conditions. And these restrictions need to be year-round. We can't just reduce nitrogen in the spring and phosphorus in the summer, because most of these systems have long residence times. You know, they have a long memory for what gets into them. So the reductions need to be year-round, even in the wintertime. You need to reduce the inputs of nutrients on a year-round basis. And lastly, the seasons are getting longer, and I think we're seeing that even here. But certainly in the northern and most southern parts of our latitudes, that is really playing a huge role. For example, in the Scandinavian countries, the ice goes out a lot earlier now, and some lakes don't have any ice on them anymore at all. So that's really extending the window of opportunity for these blooms to form, particularly with warmer surface waters. So I'll stop there and leave some room, hopefully, for some questions. I just wanted to thank all my colleagues from here, Europe, China, etc. And this wouldn't be, this work couldn't be done without all their help and input, and then the sources of support. Right now, the work in China is being done on an NSF project, Dimensions of Biodiversity. Also, the Lake Erie project is supporting our domestic work. EPA, and lastly, the Chinese government is supporting a lot of this work too, because they want to solve their problems, and we're working hard with them to help them do so. So I'll stop there and really appreciate the opportunity to talk to you, and more than glad to answer any questions.